Come again, friends. Welcome again in our in another video tutorial from microbiology with Shagnik. So this will be the continuation of our video lecture series recombinant DNA technology. And in this video lecture series, we are going to talk about isoschizomers, neoschizomers, isocaudomers. So what are they? Okay. So before going to talk about this, let's recap first. Okay. So we are talking about restriction enzymes or restriction endonuclease for a long time. Okay, so what restriction endonuclease are? Restriction endonuclease is typically evolved by bacteria to protect themselves from viral action that we have talked about. So the first restriction endonuclease that was purified, it is our HIND2. Okay, so let me this HIND2. Okay, then we have talked about what are restriction sites. So what restriction site are? Restriction site are simply a palindromic sequence of 4 to 8 base pair long that our restriction endonuclease will recognize, and they will cut this restriction site into restriction fragment, right? And this restriction cut can be of two types. One is blunt cut, one is blunt cut, and another one is sticky cut, or you can say overhang cut. Okay, blunt cut is what? Blunt cut is simply the situation in which the cleavage side is exactly on the symmetry of axis. That means blunt cut divide the restriction side into two equal part, and sticky cut divide restriction side into unequal part. So we can again divide this sticky blunt cut gives us blunt end. I have told you in my previous video, and sticky cut gives you sticky end, and this sticky end can be of two types. One is your 5 prime sticky end, another one is 3 prime sticky end. So these are the lectures that I have previously taken. Okay, so today we are going to talk about isoschizomers, neoschizomers, and isocaudomers. In the video where we have talked about restriction site, I have told you in the last that there is a term called star activity. So star activity is what? That is under non-standard condition, that is low ionic strength and a high pH, some restriction endonuclease have the ability to recognize a restriction site that is similar but not identical to their defined restriction site. And I have told you that this thing is something similar to the cross activity that we have studied in our immunology. Okay, so isoschizomers, neoschizomers, and isocaudomers are something result of this star activity phenomena. So let's talk about what isoschizomers are. So isoschizomers, let's learn this with the help of example. So suppose this is SPH1 is a restriction endonuclease, and let's revise our nomenclature scheme with the help of this. So first word simply tell us about the genus from which this enzyme has been isolated. Second two words simply tell us about the species from which it is isolated, and the Roman numbers simply tell us about the order of enzyme that has been isolated from that particular bacterium. Okay, so SPH1 has restriction site CGTACG, and this, and you have to remember that in a restriction site in which position the restriction endonuclease make a card, we will refer this by symbol, by a form called symbol. Okay, so that means that in this restriction you can say in this restriction site or restrictions recognition site, this SPH1 endonuclease will make cut in that position. Okay, and another enzyme is BBU1, and it will also recognize the same restriction sequence and make the cut in the same position. Okay, So we can say this SPH1 and BBU1 are isoschizomers. So isoschizomers are what? Isoschizomers are simply different enzymes that have the ability to recognize same restriction sequence, and they will make the cut at exactly the same location, okay, in that restriction site, okay. Now let's talk about neoschizomers. Neoschizomers is slightly different from isoschizomers. So here are two restriction endonuclease that exhibits neoschizomers. So one is SMA1, SMA1 that recognizes that particular restriction sequences, okay, and that one is XMA1 that recognizes this particular restriction site or recognition sequences, okay, and SMA1 will make the cut in that position and XMA1 will make the cut in that position, okay. So we can say that these two enzymes are neoschizomers. That means neoschizomers are different enzyme, or you can say pair of restriction endonuclease that have the ability to recognize the same sequences, but they will make the cut at different position, okay. So I hope it is clear. Now the most important one, that is your isocaudomers. What is isocaudomers are? Let's come to the example. One example is South 3 endonuclease, another one is our BAMH1, okay. So the thing is, these two endonucleases will recognize different recognition site, number one, and they will make their cut in different region, or you can say different location, but that result in the same stick end, okay. That means after the treatment with South 3 recognition site, they will exhibit the same stick end that this recognition site exhibits after treatment with BAMH1. So what there? Let's talk about. So the restriction sequence that was recognized by South 3 is NGAT, that means NGATC, and there are another nucleotides. So South 3 will make the cut here, and in opposite direction the South 3 will make the cut here, and we will have something like that. So this is stick end, as we all know. Why this is stick end? Because they do not divide this restriction site into equal position. So when they will be blunt in 1 2 3 4 5 6 7 8, okay, that means 3 4 5 6 7 8. If they will make the cut here, then your restriction site will be divided into two equal fragments. Then you can say that they will generate blunt end, but here that is not going on. So we can say this is, they will generate stick end, and the sticky end will be something like that, okay, 5 prime GATC, 5 prime GATC, okay. So this, the name of this sticky end is 5 prime GATC, and if we will again make the cut in case of BAMH1, that is in case of BAMH1, the cut, the BAMH1 will make the cut in the restriction side here, okay. So let me change the, okay. So after the treatment of BAMH1, the restriction site have two fragments, one is something like that and another one is something like that, and again there will be the same stick end, 5 prime GATC, 3 prime stick end. So BAMH1 and South 3A are the example of isocaudomers. So what isocaudomers are? Isocaudomers are simply two pairs of restriction endonuclease that will recognize different restriction site, and they will make their cut in different location, but it ultimately result in the same cohesive end, or you can say stick end, okay. So I hope it is clear. In our next video we are going to talk about frequency of restriction site, okay. That means in a a certain chromosome, what is the probability that restriction site will be present, right? So I hope this video will be helpful to you, and if it is, please like the video, share with your friends and subscribe to my channel. And if you want notes about those topics, then you can simply refer to my Facebook group named as Microbiology with Shagnik, okay. So thank you.